Okay, you guys, this is going to be a little bit of a different video than I usually do. And this video is going to be making a Punnett square on a single gene recessive. Now, this is more towards corn snakes, but can be used for any snakes. But before we get started, I'm just going to show you what some of the letters mean. So, if you have a lowercase to a lower case, that means you are homozygous recessive for that particular gene that you are plotting for, which I hope you can read that. So an example of that would be LL. If it is an upper to an upper, then the snake does lacking of that particular gene. Most people call it wild, or just doesn't possess anything. And that would be like LL. If you have an upper and a lower, this is heterozygous for that particular gene that you are looking for, and it is represented with a capital and a lower, like that. So I have made some little examples, and on the top there, before I get started, you see that it says female genetics and male genetics, and then I put the female on the top and the male on the bottom. I think scientifically there's a technical place, like the female is supposed to go one place, the male is supposed to go another, but theoretically it shouldn't really matter which way you go about it. And then down there we have the genotype and the phenotype, which I will get to in a moment. And so the first case scenario, we are going to use the diffused gene just because it's easier to do. And we're going to go a diffused to a diffused. So the female genetics would be D, lowercase d, and the male would be lowercase d. Now granted you, I should have put the word female a little bit above this, the male a little bit to the left, but it'll work regardless. So in a single gene, which is the easiest one to do, you would take one lower D from the female and transfer it above the top box. And you'll take the other lower D and transfer it to the other box on the top. And you will be repeating the same thing for the male, which I'll be representing in green, and I forgot that, so imagine that's green. That way you guys will know where it comes from. And sorry I forgot to do that. And the rest of it is just matching the two letters, which in this case it would be D from the female, purple, and then the D from the male. And then the D from the female, D from the male, D from the female, D from the male, and so on. I think you guys are getting the point and I don't have to keep repeating myself. Now granted you don't have to color code it, and I usually don't, but I find it's a little bit easier when you're first learning to have it color coded so you know exactly where things come from. And it's very easy in a single gene to know where things come from. But when you get to multi-genes and not everything is homozygous recessive and some of them are heterozygous, it gets a little messy. So it's always nice to keep track of what comes from where. Okay, so on the bottom is genotypes and phenotypes. The genotypes is actually what is in the genes. This is where something like heterozygous is going to show up. Phenotypes is what you actually see on the animal. The color, the pattern, things like that. Now, this particular case isn't really going to show the genotype very well because they're all the same genotype, which is DD, homozygous recessive for diffused. Now, what I'm going to do, one second, which I think is useful, and it's a good hobby to get into because a lot of times you're not going to be dealing with a single recessive, is to use tally marks to count how many of this genotype you specifically have. I put the tally marks in brackets. So I go 1, 2, 3, 4. And I actually write out what it is. So homo diffused. Excuse my very bad handwriting. Now in the phenotype section, all you would write down is diffused. And how many there are. And I do tally marks. And the reason, again, I do tally marks is because when you go into multi-genes, especially when you get 3 and above, writing down numbers 1, 2, 3, 4, and I'm sorry my camera isn't focusing. Thank you, camera. But like I was saying, when you get into 3 or more genes, it gets really difficult to be writing down 1, then 2, then 3, and you're pretty soon going to have your whole page covered with numbers. Tally marks are much more better and more efficient. So that is an example of a homozygous recessive diffused to a homozygous recessive diffused. I'm sorry, those are horrid tongue twisters for me. We're going to set that situation aside. Camera up just a little, if it's going to cooperate a little bit, which, thank you, camera. Okay, the female genetics this time, we're going to make the female het diffused. So that's going to be represented by an uppercase D and a lowercase d. And the male is going to be homozygous recessive diffused, which is lowercase d in green, lowercase d in green, which this is not tearing out nearly as neat as I was hoping it would be, because I keep forgetting to color code things correctly. But then you would go and trace the two genes from the female in the same exact order that it is in. So it would be uppercase D, lowercase d. Now granted, someone may say this doesn't matter, but staying organized and trying to make things the same makes things a lot easier when you're getting onto multi-genes. And then lowercase d to lowercase d. And then you would go and drag this upper D down by the purple. And then the lower D from the male, making sure you're right there. And then this lowercase d would come down to here from the female, and the lowercase d for the male would come down. And then consequently it's going to be the same results for the bottom. And the next thing you're going to do is the genotype. Now, this is actually going to be better because unlike in the first one, 100% was the same genotype and 100% was the same phenotype. So the genotype is what you start with, and the genotype is I always start with because the genotype always tells the phenotype, while the phenotype doesn't tell the genotype. That's why. There you go. I know there's a logical reason to it. It's just I'm not the best explainer to my logic sometimes, but there are only two, in this particular case, two genotypes. Two different genotypes, which would be the D, uppercase D and the lowercase d. And so I would then tally mark it, how many of those are there. And you can see from looking at the Punnett square that there are two, one right there and one right there. So I would tally mark it 2. And then I would write down het diffused. And I would also put a percentage, which in this case it'd be 50%. And I got 50% by dividing the number of that specific genotype combo by all of the combos available, and all the combos available are 4. You just count the boxes and then the number that you're looking for. Pretty easy math, not very difficult. And then I'll write down the only other combo, which is diffused. And again, I would tally mark it, write down, recessive diffuse. Of course, I do a little bit shorthand. And then the percentage. Of course, this looks better, a lot better in an Excel sheet, but I wanted to do this on pencil. I mean on paper, not on pencil, obviously. And then the two phenotypes, again, are what you actually see on the snake. It's the physical appearance. And as I can see, I actually match up with the genotype here. So a het diffuse phenotype-wise would just be a normal. Because that is all you see on the outside. There's two of those, 50%. And then there's going to be two diffused, and again, 50%. Now, it is good to know that the percentages don't always match for the phenotype and the genotype, and I can go across and assume just because this says 50% that this side will say 50%. And that's due to hets and whatnot. I'm not sure if we'll get into that specific position in this video. We may have to go to a 2+ gene to see that happening. But it's just good to know, don't ever assume that these two will always match. Now, the third scenario we're going to look at. Tripod's really difficult to move right now. But it is going to be a female, which is going to be a normal, which is big D to big D. And we're going to get a diffused male to it. So little d, little d. And again, you would go and you would write up what the female is in the top female brackets. Big D, little d for the male. And you're going to match up the male and the female, just like you did in the first two. And to be efficient, I'm just going to go ahead and put all the females down and then the males. You can do it either way, technically, but when you get into the multi-gene, I would suggest doing it one by one, and not the method I did here. With single genes, it's very easy to do it that way because the layout and the letters are only one letter, so it's no big deal. And then you go to the genotype, and in this particular scenario, it is extremely easy because there's only one phenotype, and I wish I would stop not color coding these things. Like I said earlier, I don't color code usually, so it's a little bit of a new thing to me. And all four of them are going to be like that. Het recessive for diffused, and my horrid handwriting and spelling, and phenotype is going to be normal. 1, 2, 3, 4, 100%. And 100% for over here. If you don't see it, that's 100%. So far we have done a het to a homorecessive refused, a two diffused homozygous recessives, and a normal to a homozygous recessive diffused. Now what we're going to do this time and is going to be a female normal to a male het diffused. And you couldn't see that. Yay, because I forgot to move the tripod. Oh well, so you would again drag females, one letter from each female onto the box, and one letter from each male down to the box. And for time's sake, I'm just going to do this really quick. But when you're doing it this way, to watch yourself, to make sure you do not transfer the wrong letter that represents things, because otherwise you're going to be in quite the situation. It really doesn't take much to mess up the whole scenario. So as you can see, we have two normals and two hets just by looking at that. you would again write the genotypes down first. And there are 2 of those, so 2 tally marks, and then normal. Just write normal 150%. And then D, there is 2 of those, and that's het diffused, het D 50%. And then I would have normal, all 4 of them. And this is where it comes in what I mentioned earlier, not to assume that these 2 percentages always match, all these percentages match up. Because on the outside, the phenotype, which is again what the snake looks like on the outside, will all appear normal. Because het does not appear on the snake. For it to appear on the snake, the snake in question has to have both copies of the gene, which would be 2 lowercase d's. And a het only possesses one of those. And the upper D just presents wild type normal / doesn't have that particular gene you're searching for. The last case scenario that I'm going to at least be doing on this session is the het to a het scenario. So a het diffused to a het diffused. So big D to the little d, big D, and little d. And for time's sake, again, I'm just going to do this because it takes quite a bit of time to do each square by itself. Little d, little d. And as you can see right now, the situation has got quite a bit more complicated than when we first started. So the first situation we have to plot down the genotype is big D to big D. Now there's only one of those, so I would only have 1 tally mark there. And that is a normal. And that is 25%. The second type is big D to little d. Now granted because the big D's both come from different parents, I'm just going to write it whichever color I desire, because the colors are only there to show you where the letters came from. So big D, little d. And there are 2 of those. And that's het diffused. That's 50% of them. And then the last combo that you can see is these 2 little d's. I'm going to apologize if my camera is going out of focus. There is nothing I can do about it. It's already on a tripod. So, don't know what else to say about that. And that is homo recessive diffused, 25%. And the phenotype is going to be a lot more complex than the previous ones. Because normals and hets both appear as normals, there are actually 3 of those. Which would be 75%. And there would be only 1 diffused. I apologize for my handwriting, it's not good. It's never been good and it's never going to get good. And before I'm going to end this video, I'm going to talk about these percents, because these percents are not going to say that 25% of your clutch is going to be normal, 50% is going to be het, and 25 is going to be diffused. This is the probability of each single egg being the specific genotype. In breeding of corn snakes, this is really what's looked at, is the genotype because of the het. Normal is the het. Homozygous recessive diffused, sorry, it's a tongue twister. It's obvious when the snake comes out of the egg if it has that. But because these two look exactly the same, unless you know the parents' genetics and you prove it out through breeding trials, you're not going to know if it's normal or if it's recessive. It's het recessive. So these are just probabilities. And this is where you get the percentage chance probabilities of possible hets. This, if they want to say het, this particular clutch, if they would say 50% het, that is exactly where that 50% comes from. Let me check to see if I can find the 66% het. 100% het. 100%. That's a 50. The 60%, 66% het, I believe, would come from the and I'm looking really bad right now because I can't find exactly where that situation is arising, but the 66% nonetheless, I may not have drawn it out, I may have. I'm not going to go look through the pile anymore, but it's the same thing. Instead of 50%, it's more like 66% situation. So yeah, that is that. Unless this is the 66%. Now I'm rambling. So technically I've written 50% down, but if you take 25% off. Yeah, this might actually be the 66% chance. Either way, I'm not going to get into resemble which one's 66 and which one's not 66%. But because obviously if you take this away, 2 out of 3 is 66%. So no, I was wrong. This is the 66% scenario. That is why I could not find it in the pile, because I was on it. I forgot to take the diffused out. So it is no longer 2 out of 4. It is 2 out of 3, and 2 out of 3 is 66%. So before anyone writes in the comment section, you're on it. I found it. And a 50% would be, 1 second, because this would be 100% het, because it would be, no it wouldn't, that would not be a 100% het. Damn it, I'm doing really bad right now. This went all well until I started going through these piles and they're out of my order. This would be the 50% chance of being a het. 100% het. 100% het. And all diffused with 100% het. This would repeat itself because it'd be obvious it came out as diffused, and the only other scenario is a het. So I'm really sorry, guys, about the confusion. I'm not the best at the percentages, especially when I have it down the genotypes and it says 50% and not exactly what's meeting up with snake logics. So yeah, I shall bid you adieu, and I apologize for the little bit of mix-up on the percentages.